The forest floor is soft beneath my feet, cushioned by decades of accumulated leaf litter, slowly decomposing into soil. I'm walking through an old-growth woodland in the Pacific Northwest during peak autumn, when the fungal fruiting season reaches its crescendo and the forest floor erupts with mushrooms in astonishing diversity. The air is cool and damp, heavy with the earthy scent of decomposition, not the unpleasant smell of rot, but the rich, complex aroma of organic matter being broken down and recycled, the fundamental process that sustains all terrestrial life. I've come here as a mushroom hunter, though my hunt is more about observation and understanding than harvest. I carry a woven basket, a small knife, field guides, and a hand lens for examining details too small for the naked eye to resolve. But mostly, I carry curiosity about the hidden world beneath my feet, the vast mycelial networks that connect trees, break down dead wood, form partnerships with plant roots, and occasionally produce the visible fruiting bodies we call mushrooms. Most people think of mushrooms as plants, or at least as plant-like organisms, but fungi are actually more closely related to animals than to plants. Like animals, they're heterotrophs. They can't photosynthesize and must obtain energy by consuming organic matter. Like animals, they store energy as glycogen rather than starch. At the cellular level, fungal cells have characteristics that align them more closely with animal cells than plant cells, though they diverged from our common ancestor over 1 billion years ago and have evolved unique features that suit their particular lifestyle. What we call a mushroom, the visible fruiting body pushing up through leaf litter or emerging from tree bark, is actually just the reproductive structure of a much larger organism. The main body of a fungus is the mycelium, a network of thread-like filaments called hyphae that spread through soil, wood, or whatever substrate the fungus inhabits. These networks can be enormous. The largest known organism on Earth is a honey fungus, Armillaria ostoyi, in Oregon's Blue Mountains, with a mycelium spreading across nearly 10 square kilometers, estimated to be thousands of years old. The mycelium is where the real action happens. These microscopic threads explore their environment, secreting enzymes that break down complex organic molecules into simpler compounds the fungus can absorb. They transport nutrients and water, sometimes over considerable distances within the network. They form connections with plant roots and other fungi, creating underground networks of exchange and communication that scientists are only beginning to understand. The mushrooms we see above ground exist primarily to disperse spores, the fungal equivalent of seeds, but they represent just a tiny fraction of the organism's total mass and complexity. I kneel beside my first find of the day, a cluster of honey mushrooms, Armillaria malea, growing at the base of an old Douglas fir. These are beautiful mushrooms, with honey-colored caps scattered with darker scales, white stems with distinctive rings, and gills that extend slightly down the stem. But they're also parasites and decomposers, capable of killing living trees while simultaneously breaking down dead wood. The relationship is complex. Sometimes they cause serious damage to forest trees, earning them a reputation as pests among foresters. But they also play crucial ecological roles, helping to break down dead wood and creating gaps in the forest canopy that allow new trees to establish. The honey mushroom cluster probably represents a single individual, one genetic organism that has colonized this tree and is producing multiple fruiting bodies. If I were to follow the mycelium from this location, I might find it extending to other trees in the area, possibly connected to that massive Oregon individual across a network spanning miles. Fungi don't respect the boundaries we use to define individual organisms. A mycelial network can fragment and continue as separate individuals or separate networks, confuse if they're genetically compatible, creating organisms that challenge our understanding of what individual means. I move deeper into the forest, my eyes adjusting to the patterns of mushroom shapes and colors among the leaf litter. Once you develop the search image, the ability to recognize mushroom forms among the visual complexity of the forest floor, they seem to appear everywhere. A flush of delicate mycena pushing up through moss on a rotting log, their translucent caps glowing like tiny lanterns. A massive shelf fungus extending from a standing dead tree, its bracket-like form covered in pores rather than gills, tough and woody rather than soft and fleshy. The diversity is astounding. Scientists estimate there might be between 2.2 and 3.8 million fungal species globally, though only about 150,000 have been formally described and named. For every plant species, there might be 6 fungal species. Fungi occupy nearly every terrestrial habitat and many aquatic ones. They range in size from single-celled yeasts to organisms covering thousands of acres. They've evolved to exploit every conceivable source of organic carbon, living plants, dead plants, animal dung, other fungi, even living animals, including humans. I find a perfect specimen of what appears to be a matsutake, Tricholoma matsutake, one of the most prized edible mushrooms, known for its distinctive spicy aromatic scent. I don't pick it. Matsutake populations are declining in many regions due to overharvesting and habitat change, and I'm here to observe rather than harvest. But I kneel close, inhaling its characteristic fragrance, examining the way it pushes up through the duff, partially hidden, as is typical for this species. Matsutake form mycorrhizal relationships with certain trees, mutualistic partnerships where the fungus colonizes tree roots, helping the tree absorb water and nutrients while receiving sugars the tree produces through photosynthesis. Mycorrhizal relationships are among the most important on Earth, though they remained largely unknown until relatively recently. Most land plants, estimates suggest 90% or more, form mycorrhizal partnerships with fungi. The fungal partner extends the plant's root system enormously, with hyphae exploring volumes of soil far beyond what roots alone could access. The greatly increased surface area allows more efficient uptake of water and minerals, particularly phosphorus, which is often limiting despite being abundant in soil, because it exists primarily in forms plants can't directly access. The fungi can enzymatically liberate phosphorus from organic matter or mineral forms, making it available to their plant partners. In exchange for this service, the plant provides the fungus with carbohydrates, sugars produced through photosynthesis. This might seem like a simple trade, but the relationship is far more sophisticated than just nutrient exchange. Research in recent decades has revealed that mycorrhizal networks connect multiple plants, often of different species, creating what some scientists call the wood wide web. Through these networks, trees can share resources with larger, well-established trees, sometimes supporting younger or struggling neighbors by transferring carbon and nutrients through the fungal connections. Mother trees, the large ancient individuals that anchor forest ecosystems, can recognize their own offspring through these networks and preferentially support them with additional resources. The networks also facilitate communication. When a tree is attacked by insects, it can release chemical signals that travel through the mycorrhizal network, warning neighboring trees of the threat and allowing them to activate defensive compounds before the insects reach them. Plants under stress can signal through the network, potentially receiving resources from healthier neighbors. The fungi themselves benefit from connecting multiple plants. If one plant dies or becomes less productive, the fungus maintains its other connections, diversifying its carbon sources the way an investor diversifies a portfolio. I'm walking now through a section of forest dominated by western hemlock and western red cedar, both of which form mycorrhizal relationships with numerous fungal species. The forest floor here is a tapestry of mushrooms, and I take time to examine them carefully, photographing specimens, noting their characteristics, trying to identify them using my field guides. Some are relatively easy. The bright red caps with white spots of fly agaric, Amanita muscaria, are unmistakable, though the white spots can wash off in rain, leaving smooth red caps that are less distinctive. Despite its fairy tale appearance, this is a toxic mushroom, containing psychoactive compounds that have been used ceremonially in some cultures, but can cause serious poisoning if consumed without proper preparation. Other mushrooms are more challenging to identify. The little brown mushrooms, or LBMs as mycologists call them, dozens of species that look superficially similar, requiring microscopic examination of spore characteristics or even DNA sequencing for certain identification. The white-spored species that could be any of several genera, some edible and choice, others poisonous, and potentially deadly. Mushroom identification requires careful attention to multiple characteristics: cap shape, color, and texture, gill attachment, spacing, and color, stem features including rings, bulbs, or surface texture, spore print color, habitat, and associated plants, and often smell and taste. Though tasting unknown mushrooms is done cautiously, sampling tiny amounts and spitting them out, rather than swallowing. The stakes for accurate identification can be high. Some mushrooms are deadly poisonous, containing toxins that cause liver failure or kidney damage, often with a delayed onset that makes treatment difficult. The death cap, Amanita phalloides, responsible for the majority of fatal mushroom poisonings worldwide, can look similar to several edible species and tastes pleasant, giving no warning of its toxicity. A single mushroom can contain enough amatoxins to kill an adult human, and there's no antidote. Treatment is supportive care while hoping the liver can regenerate faster than the toxins destroy it. But many mushrooms are not only safe, but nutritious and delicious. Chanterelles, with their golden color, fruity aroma, and exquisite flavor, are prized by chefs and foragers. Porcini and other boletes offer meaty texture and rich, earthy taste. Lion's mane, with its cascading white spines, has both culinary and potential medicinal properties. Oyster mushrooms growing on dead wood are easy to identify and cultivate. Foraging for mushrooms combines the satisfaction of finding food with the intellectual challenge of identification and the sensory pleasure of cooking and eating something you've gathered yourself. I find a beautiful cluster of chanterelles, Cantharellus formosus, pushing up through the moss beneath a Douglas fir. Their golden orange color is distinctive, and they have false gills, ridges rather than true gills, running down the stem in forking patterns. They smell faintly of apricots, another identifying characteristic. Chanterelles are mycorrhizal partners with conifers, so their presence indicates a healthy fungus-tree relationship underground. I photograph them, but leave them in place. I have no collection permit for this protected forest area, and besides, I'm more interested in understanding their ecology than in harvesting them. The mycorrhizal relationship chanterelles form with their tree partners has been studied extensively because these mushrooms are economically important. They're harvested commercially in many regions and can sell for high prices in markets. But chanterelles can't be cultivated using conventional mushroom farming techniques because they require living tree roots as partners. All attempts to grow them apart from their tree hosts have failed, illustrating how obligate and specific these relationships are. You can't simply grow chanterelle mycelium on compost or logs the way you can with oyster mushrooms or shiitake. The fungus needs its tree partner, and the partnership takes years to establish, which is why sustainable wild harvesting is the only current source for these mushrooms. The sound of the forest has a particular quality in autumn. Less birdsong than in spring or summer, but more sounds of small animals preparing for winter. I hear the chattering of a Douglas squirrel, probably harvesting cone seeds and storing them for the lean months ahead. The soft rustle of a varied thrush foraging in leaf litter, flipping leaves to expose invertebrates. And underneath it all, the white noise of wind through conifer needles, rain dripping from canopy to understory to ground, the distant sound of a creek swollen with autumn rains. I come across a massive log, probably from a Douglas fir that fell decades ago, now deeply involved in the process of decomposition. The wood is soft and punky, easily compressed when I press on it, shot through with mycelial threads that appear as white strands running through the darker wood. The log is also sprouting mushrooms from every surface, shelf fungi and overlapping tiers, small clusters of delicate species, and one spectacular clump of what I believe is chicken of the woods, Lataparus sulphureus, with its brilliant orange and yellow shelf-like formations extending in thick, fleshy brackets. This log represents a crucial phase in forest nutrient cycling. When a tree falls, it contains tons of organic matter, cellulose, lignin, and other complex molecules that represent decades or centuries of captured carbon and absorbed nutrients. But this material is locked up in forms that most organisms can't access directly. Fungi are among the few organisms with the enzymatic machinery to break down lignin, the complex polymer that gives wood its strength and durability. Through a combination of mechanical penetration and chemical degradation, fungi slowly dismantle the wood structure, liberating nutrients and carbon that become available to other organisms. The decomposition of a single large log can take decades, proceeding through distinct stages as different species of fungi and other decomposers succeed one another. Early colonizers are often pioneer species that can tolerate the relatively harsh conditions of fresh wood, low nitrogen content, chemical defenses the tree produced while alive, competition with other organisms. These pioneers begin the breakdown process, making the wood softer and more hospitable for the next wave of colonizers. Later stage fungi specialize in thoroughly degraded wood, extracting resources from increasingly recalcitrant material until the log is fully integrated back into the soil. Throughout this process, the log serves multiple ecological functions beyond just nutrient cycling. It provides habitat for countless invertebrates, beetles, ants, termites, centipedes, spiders, and many others that bore into the wood, nest within it, or hunt among its crevices. These invertebrates become food for salamanders, shrews, birds, and other predators. The log acts as a moisture reservoir, staying damp even during dry periods and providing critical habitat for moisture-dependent species. It serves as a nurse log for seedlings, young trees that germinate on its surface, their roots eventually wrapping around and through the log as it decomposes, eventually standing on stilted roots once the log has fully decomposed beneath them. I examined the chicken of the woods more closely. This species is unusual in that it's a parasite that attacks living trees, but it continues decomposing wood after the tree dies, serving dual roles. The bright orange and yellow coloration is striking against the dark, decomposing wood. Unlike many mushrooms with delicate, ephemeral fruiting bodies, chicken of the woods produces substantial, fleshy brackets that can weigh several pounds and persist for weeks. Young specimens are edible and have a texture and flavor that some say resembles chicken, hence the common name, though as with all wild mushrooms, proper identification is critical. There are toxic lookalikes, and even true chicken of the woods can cause digestive upset in some people. The fungal diversity on this single log is remarkable. I count at least 15 different species producing visible fruiting bodies, and there are undoubtedly dozens more present as mycelium within the wood, but not currently fruiting. Each species has its niche. Some specialize in breaking down cellulose, others focus on lignin, some prefer the sapwood near the bark, others colonize the heartwood deeper inside. Some are competitive, secreting compounds that inhibit other fungi. Others are more cooperative, or at least tolerant of sharing the resource. The community of fungi within a decomposing log is as complex and dynamic as any above-ground ecosystem, just less visible to casual observation. I settle onto a dry portion of the log to rest and observe, pulling out a small hand lens to examine some of the smaller mushrooms more closely. One particular species catches my attention. Tiny mushrooms no more than a few millimeters tall, growing in dense clusters on the bark. Through the lens, I can see they have perfect miniature mushroom form, caps, stems, gills, but at a scale that makes them easy to overlook without close observation. These might be Mycena species, a diverse genus of small, delicate mushrooms that often grow on dead wood or among moss. Some Mycena are bioluminescent, glowing with a soft green light in darkness, though I'd need to return at night to see if these particular specimens have that property. Bioluminescence in fungi is a fascinating phenomenon that occurs in several dozen species worldwide. The light is produced through a chemical reaction similar to that in fireflies. A substrate called luciferin is oxidized by an enzyme called luciferase, releasing energy as light. But while firefly bioluminescence serves clear communicative functions, attracting mates, deterring predators, the function of fungal bioluminescence is less obvious. Some theories suggest it attracts insects that help disperse spores, or that it's simply a metabolic byproduct with no adaptive function, or that it might deter organisms that would eat the mushroom. The truth likely varies by species, and researchers are still investigating the ecology of glowing fungi. The forest around me is a three-dimensional habitat extending from deep in the soil through multiple canopy layers. I'm most aware of the ground layer where I'm sitting, but above me, fungi inhabit the canopy as well. Species growing on living bark, in accumulated organic matter in branch crotches on dead branches, even as endophytes living inside healthy leaves without causing obvious harm. Below me, the soil is shot through with mycelial networks extending meters in every direction, most belonging to mycorrhizal species forming partnerships with tree roots, but many others decomposing organic matter in the soil or parasitizing soil invertebrates. One group of particularly remarkable fungi are the cordyceps, parasites of insects, and other arthropods. These fungi infect their hosts, often altering the host's behavior in ways that benefit the fungus. The famous zombie ant fungus, Ophiocordyceps unilateralis, causes infected ants to climb to elevated locations and bite down on vegetation, then kills them and grows a fruiting body from the ant's head that releases spores from this elevated position, maximizing dispersal. Different cordyceps species specialize in different hosts. There are cordyceps that infect caterpillars, beetles, spiders, even other fungi. Some cordyceps have been used in traditional medicine for centuries and are now being investigated for potential pharmaceutical compounds. The relationship between fungi and insects is complex and multifaceted, not just parasitism, but also mutualism and commensalism. Many insects cultivate fungi deliberately, carrying fungal spores or mycelium and inoculating suitable substrates, then feeding on the fungi they've cultivated. Leafcutter ants are famous for this. They harvest fresh leaf material, carry it underground, chew it into a pulp, and use it as substrate for growing fungus gardens that serve as their primary food source. The fungi they cultivate belong to the genus Leucoagaricus and are found nowhere else. They've been domesticated by ants for millions of years and are now entirely dependent on their ant partners. Bark beetles and ambrosia beetles also cultivate fungi, carrying fungal partners in special structures on their bodies and inoculating trees with these fungi when they bore into wood. The fungi help break down wood, making it more digestible for beetle larvae and produce nutrients that the beetles require. Some of these beetle-fungus partnerships are devastating to forest trees. Dutch elm disease and various bark beetle outbreaks that have killed millions of trees in western North America involve fungi carried by beetles, with the fungi often being the actual killing agent. I stand and continue my walk, following no particular path, just moving slowly through the forest, stopping frequently to examine interesting specimens. The ground beneath my feet is incredibly complex when you consider it from a fungal perspective. Every step lands on a surface that's laced with mycelial threads, millions of high-fall tips exploring tiny soil pores, colonizing organic matter, forming connections with plant roots. Soil fungi contribute enormously to soil structure, producing sticky compounds that bind soil particles together into aggregates that resist erosion and facilitate water infiltration. The soil food web is staggeringly complex, with fungi playing central roles at multiple trophic levels. As decomposers, they break down dead organic matter, releasing nutrients that can be absorbed by plants. As mycorrhizal partners, they connect to plant roots and trade nutrients for photosynthetic sugars. As prey, they're consumed by specialized fungivores, nematodes, springtails, mites, and other tiny invertebrates that graze on mycelial threads or specialize in consuming spores. Some soil bacteria prey on fungi, secreting enzymes that break down fungal cell walls, and some fungi are themselves predators, forming specialized structures that trap nematodes, microscopic roundworms abundant in soil, then consuming them to supplement their nitrogen intake. These predatory fungi have evolved remarkable trapping mechanisms. Some form adhesive networks that snare passing nematodes like sticky flypaper. Others form constricting rings. When a nematode passes through the ring, contact triggers the ring cells to rapidly inflate, constricting around the nematode and holding it fast while hyphae penetrate its body and consume it from within. Still others form adhesive knobs that stick to nematodes on contact, or produce toxins that paralyze their prey. These strategies allow fungi to capture mobile organisms and extract nutrients, particularly nitrogen, that would otherwise be unavailable from their substrate. I find a cluster of purple coral mushrooms, Clavaria zollingeri, pushing up through the leaf litter, their delicate branching forms resembling tiny purple corals. These are saprobic species, meaning they obtain nutrients by decomposing dead organic matter rather than forming partnerships with plants. Coral fungi are common in forest floors, but often overlooked because of their unusual form. They don't look like stereotypical mushrooms with caps and stems, instead producing upright branch structures that release spores from their surfaces. Some species are edible, others are too small or tough to be worthwhile, and a few are mildly toxic. The diversity of fungal forms is extraordinary. Mushrooms are just one morphology among many. Puffballs produce spherical fruiting bodies that release spores through openings when mature or when disturbed, a satisfying puff of brown smoke that gives them their name. Earth stars are related to puffballs, but have an outer layer that splits and peels back in star-like rays, elevating the central spore-bearing structure. Stinkhorns produce phallic or lattice-like structures coated with foul-smelling spore-bearing slime that attracts flies, which then disperse the spores. Bird's nest fungi create tiny cup-shaped structures containing lentil-sized eggs full of spores. Raindrops hitting the cups splash the eggs out, projecting them considerable distances. These diverse forms represent different solutions to the fundamental challenge of spore dispersal. Gilled mushrooms position spores on vertical gill surfaces and rely on air currents to carry spores away once they're released. Boletes have pores instead of gills, providing similar function with different geometry. Shelf fungi on trees produce pores on the underside of horizontal brackets, positioning spores to fall free of the tree. Puffballs trap spores until a dispersal event, rain, animal contact, wind, releases them all at once. Each morphology reflects adaptations to specific dispersal challenges and opportunities. The sheer number of spores fungi produce is staggering. A single giant puffball might contain 7 trillion spores. A large mushroom can release billions of spores over its few-day lifespan. This massive reproductive output is necessary because the vast majority of spores never successfully establish. They land in unsuitable locations, fail to find appropriate substrate, are consumed by fungivores, or are outcompeted by other organisms. Fungal reproduction is a numbers game, producing astronomical quantities of propagules in hopes that a tiny fraction will succeed. I notice my basket contains a small collection now, not mushrooms I've picked, but interesting finds I've carefully photographed and documented before leaving in place. A remarkably perfect specimen of an amanita showing all identifying features. An unusual slime mold, technically not a fungus, but often studied by mycologists, in its plasmodial stage looking like a spreading mass of yellow foam slowly creeping across rotting wood. A beautiful example of lichen, which is actually a partnership between fungus and algae, or cyanobacteria, forming crusty patterns on a rock surface. Lichens deserve special attention because they demonstrate the extraordinary capacity for symbiosis that fungi possess. The fungal partner, usually an ascomycete, provides structure and protection, while the photosynthetic partner, green algae or cyanobacteria, produces sugars through photosynthesis. Together, they create an organism that can colonize bare rock, survive extreme desiccation, tolerate temperature extremes, and photosynthesize whenever moisture is available. Lichens are pioneer species on bare rock, slowly dissolving minerals and accumulating organic matter, beginning the process of soil formation that eventually allows other plants to establish. Lichens grow extremely slowly. Some species add just millimeters per year, which means large lichens can be very old. With some specimens estimated at thousands of years old, they are also extremely sensitive to air pollution, particularly sulfur dioxide, making them useful bioindicators. The presence or absence of certain lichen species tells you about air quality. In heavily polluted areas, sensitive species disappear, leaving only the most tolerant species or no lichens at all. As air quality improves following pollution controls, sensitive species gradually recolonize, providing a living record of atmospheric conditions. The forest is dimming now as afternoon transitions toward evening, the light taking on a softer quality as it filters through the canopy. Shadows lengthen and the temperature drops slightly, becoming distinctly cool, though not yet cold. This is the daily rhythm of the temperate forest: moderate temperature swings, reliable moisture, a long growing season compared to higher latitudes, but with distinct seasonal changes that structure ecosystem processes. I'm approaching a section of forest where a large tree recently fell, creating a gap in the canopy and allowing light to reach the forest floor in quantities unavailable in the closed canopy sections. The increased light triggers a pulse of growth. Understory plants that were surviving in deep shade suddenly have access to abundant light and respond with rapid growth. Seedlings that were biding their time, waiting for such an opening, now race upward to claim the gap. This is forest dynamics in action, the cycle of growth, death, decay, and regeneration that operates constantly, creating a mosaic of different aged patches across the landscape. The gap also affects fungal communities. Increased light and temperature at the forest floor speed decomposition rates. The fallen tree becomes a massive resource pulse. Tons of dead wood suddenly available to decomposer fungi. Species specialized in colonizing fresh fallen wood arrive quickly, some dispersing as spores from nearby sources, others already present in the tree as endophytes that were living benignly inside the living wood and now shift to aggressive decomposition of the dead tissue. Over the years and decades it takes for this tree to fully decompose, dozens of fungal species will participate in its breakdown, each playing its part in returning the tree's accumulated carbon and nutrients to the ecosystem. Sitting near the fallen tree, I take out my field notebook and begin sketching some of the mushrooms I've encountered today, trying to capture the details that will help me remember them. The way gill attachment varies between species, the distinctive colors and patterns, the habitat associations. Drawing forces careful observation, making you notice features you might otherwise overlook. The subtle curve of a cap margin, the way scales arrange on a stem, the branching pattern of false gills. These details matter for identification and tell stories about ecology and evolution. Mushroom taxonomy has been revolutionized by DNA sequencing in recent decades. Species that look similar might be genetically distant, while others that appear quite different might be closely related. Genera have been reorganized, family boundaries redrawn, and countless new species discovered through genetic analysis of specimens that had been sitting in museum collections for decades, misidentified or lumped with other species. The rate of discovery is actually accelerating as sequencing becomes cheaper and more accessible. Mycologists are finding that fungal diversity has been vastly underestimated. Environmental DNA analysis, extracting and sequencing DNA directly from soil samples, reveals the presence of numerous fungal species that never produce visible fruiting bodies, or that fruit so rarely they've never been collected. These cryptic species might be mycorrhizal specialists, tiny decomposers, or organisms with life strategies we don't yet understand. Every gram of forest soil likely contains DNA from hundreds of fungal species, most of which have never been formally described by science. We're only beginning to understand the true scope of fungal diversity. I notice movement on the fallen tree and move closer to investigate. A small slug is grazing on the surface of a mushroom, leaving a trail of slime and a distinctive feeding pattern. Neat semicircular bites taken from the cap edge. Many animals feed on mushrooms despite toxins that deter most vertebrates. Slugs and snails seem largely immune to mushroom toxins, as are many insects. Deer browse mushrooms selectively, apparently able to distinguish toxic from edible species. Squirrels harvest and cache certain mushrooms, perhaps using compounds in them to help survive winter, or perhaps appreciating their nutritional value. The relationship between fungi and animals extends beyond simple consumption. Some animals use mushrooms medicinally. Bears have been observed seeking out particular fungal species when ill or injured, and chimpanzees consume certain mushrooms outside their normal diet, possibly for parasite control. The antimicrobial and antiparasitic properties of many fungal compounds suggest that self-medication through mushroom consumption might be widespread among animals, though proving intentionality is challenging. Fungi have provided numerous pharmaceutical compounds to human medicine. Penicillin, the first widely used antibiotic, is produced by the fungus Penicillium. Many other antibiotics are fungal products. Organisms that spend their lives competing with bacteria in soil have evolved impressive chemical arsenals for bacterial suppression. Statins, widely prescribed for cholesterol management, were first isolated from fungi. Cyclosporine, essential for preventing organ transplant rejection, comes from a fungal source. Ongoing research continues to discover new bioactive compounds in fungi with potential applications ranging from cancer treatment to antiviral therapy. Traditional medicine systems worldwide have long recognized fungi's medicinal properties. Chinese medicine uses dozens of fungal species, some like reishi, Ganoderma lucidum, and turkey tail, Trametes versicolor, now being investigated by Western pharmacology and showing promising immune-modulating effects. Chaga, Inonotus obliquus, a parasitic fungus that grows on birch trees, has been used in northern European folk medicine for centuries and is now being studied for antioxidant and potential anti-cancer properties. Lion's mane, Hericium erinaceus, shows promise for nerve regeneration and cognitive support. But skepticism is warranted. Many claims about medicinal mushrooms aren't well supported by rigorous research, and the supplement industry often markets products with insufficient evidence or quality control. Some compounds that show activity in laboratory studies don't translate to clinical benefits. Others might be present in whole mushrooms, but not in extracts or supplements. And as with any biologically active substance, there's potential for side effects and interactions with medications. The promising thing is that serious scientific research is increasingly being conducted, helping separate genuine therapeutic potential from marketing hype. The evening light is fading faster now, and I realize I should begin making my way back toward the trailhead before darkness makes navigation difficult. But I'm reluctant to leave. There's a peace in this forest, a sense of being surrounded by ancient processes, part of a living system that extends through soil and wood and air, connecting everything into one vast network of relationships. The trees around me aren't solitary individuals, but nodes in a network, linked underground by mycorrhizal highways, sharing resources and information, supporting each other through partnerships millions of years old. As I walk, I think about how profoundly fungi have shaped terrestrial life. Plants couldn't have colonized land without fungal partners. The earliest land plants, appearing roughly 475 million years ago, already had mycorrhizal associations, and fossil evidence shows these relationships were crucial for extracting nutrients from primitive soils that lacked the structure and fertility of modern soils. Fungi helped build those modern soils, breaking down rock and organic matter, creating the substrate that supports today's forests and grasslands. Fungi also shaped plant evolution. The arbuscular mycorrhizal relationship, the most ancient form involving fungal structures that penetrate root cells, is nearly universal among plants, including most crops. The ectomycorrhizal relationship, where fungi form sheaths around root tips without penetrating cells, evolved more recently and is characteristic of many forest trees, including pines, oaks, beeches, and birches. These different relationships involve different fungal groups and provide different benefits, but all represent evolutionary partnerships that have become so fundamental that neither partner thrives without the other. I pass a massive western red cedar that must be 500 or 600 years old, its trunk several meters in diameter, its bark deeply furrowed and shaggy. This individual tree was already well established when European explorers first reached this coast. It's survived countless storms, droughts, fires, disease outbreaks, and insect attacks. Beneath it, extending through the soil for meters in every direction, is a network of mycorrhizal fungi, connecting this tree to dozens or hundreds of others, sharing resources, transferring information, creating a biological internet that predates human technology by hundreds of millions of years. The concept of the forest as a superorganism, a network of interconnected individuals functioning as an integrated whole, challenges traditional notions of competition as the primary driver of ecology. While competition certainly occurs, cooperation through mycorrhizal networks and other mechanisms is equally important. Old trees support young trees. Trees under stress receive resources from healthier neighbors. The forest is both community and individual, both competitive and cooperative, both fragmented and united. These apparent contradictions resolve when you recognize that different processes operate at different scales and in different contexts. I find myself at the edge of a small stream cutting through the forest, its clear water flowing gently over a bed of smooth stones. Even here, fungi are present, aquatic species that have adapted to life in water, decomposing submerged leaves and wood. Some produce spores with appendages that help them attach to substrates or facilitate dispersal in flowing water. Others form unique fruiting bodies adapted to aquatic conditions. Fungi colonized water before plants colonized land, and while most fungal diversity is now terrestrial, aquatic species remain important in freshwater ecosystems. The stream itself is part of the nutrient cycling I've been observing throughout the forest. Water flowing through soil carries dissolved organic carbon, much of it produced by fungi, breaking down organic matter and releasing soluble compounds. This dissolved organic carbon supports aquatic food webs, providing energy for bacteria and other microorganisms that form the base of the aquatic food chain. The connection between terrestrial and aquatic systems is intimate. What happens in the forest directly affects the stream, and organisms in the stream depend on resources generated by forest processes. I noticed something interesting on a submerged log, what appears to be a jelly fungus, a group of fungi that produce gelatinous fruiting bodies that can rehydrate after drying out. When moist, they're soft and jelly-like, but when dry, they shrink to hard, inconspicuous crusts that are easy to overlook. This adaptation allows them to survive variable conditions. They can fruit during wet periods, then persist through dry spells, then resume activity when moisture returns. It's an elegant solution to the challenge of living in an environment where water availability fluctuates dramatically. Jelly fungi belong to a fascinating group called Heterobasidiomycetes, characterized by their unique cellular structures and life cycles. Many are wood decomposers, some are parasites of other fungi or plants, and their gelatinous texture sets them apart from the firmer flesh of typical mushrooms. While few are considered choice edibles, some species are consumed in Asian cuisine, prized for their texture rather than flavor. Wood ear, Auricularia auriculajudae, is cultivated commercially and widely available in Asian markets. Sitting on a rock beside the stream, I take out a small thermos of tea I've brought and pour a cup, savoring its warmth and the herbal aroma rising with the steam. The sound of flowing water creates a peaceful background, a constant gentle rushing that masks other forest sounds. Water has always been associated with contemplation and peace in human cultures, perhaps because flowing water creates white noise that drowns out distracting sounds, or because water sources are places where animals gather and thus benefit from calm human presence, or simply because water is essential to life and our brains recognize its importance at a fundamental level. The relationship between water and fungi is complex. Fungi require water for growth. Their hyphae need moisture to extend. Their enzymes function in aqueous environments. Their spores often require moisture for germination. But fungi are also remarkably adapted to water stress. Many species can survive extreme desiccation, shutting down metabolism when dry and resuming activity when moisture returns. Some produce drought-resistant structures, sclerotia, which are compact masses of hardened mycelium that can survive harsh conditions and germinate when favorable conditions return. Water availability structures fungal communities in predictable ways. Wet environments favor fungi adapted to continuous moisture. Many of the jelly fungi, some mushroom species that require high humidity, and aquatic species. Dry environments favor fungi with drought tolerance, desert species that fruit only after rare rains, fungi that produce protected spores, species that grow in protected microhabitats where moisture persists. Between these extremes, temperate forests like this one support incredible fungal diversity because they provide both reliable moisture and seasonal variation, creating numerous niches. I finish my tea and rinse the cup in the stream, watching the water flow over my hands, cold and clean, carrying its dissolved cargo of organic compounds toward larger streams, eventually reaching rivers and the ocean. Even at this microscopic chemical level, fungi are influencing what flows downstream. The specific compounds they release as they decompose organic matter, the minerals they solubilize from rock, the structural changes they cause in soil that affect water movement and filtration. Continuing my walk, now following the stream for a while, I notice how the riparian zone, the area immediately adjacent to water, has its own characteristic vegetation and fungal communities. Willows and alders dominate, both nitrogen-fixing species that enrich soil. The understory is lush with moisture-loving plants, ferns, devil's club, salmonberry, and the fungi here include species specialized for riparian conditions, those that tolerate periodic flooding, species associated with specific riparian tree species, and wood decomposers adapted to the distinctive conditions of waterlogged wood. The concept of microhabitats is crucial for understanding biodiversity. A forest isn't a uniform environment, but a complex mosaic of different conditions, wet and dry areas, sunny and shaded spots, different soil types, various substrates. Each microhabitat supports somewhat different communities of organisms, including fungi. This environmental heterogeneity is one reason forests support such high biodiversity, not because any one spot is extraordinarily diverse, but because the landscape includes so many different spots, each with its own assemblage of species. Managing forests for biodiversity means maintaining this heterogeneity, preserving different age classes of trees, retaining dead wood in various stages of decay, protecting riparian areas, allowing natural disturbances like small fires and wind throws that create patches at different successional stages. Simplified forests, like plantations where all trees are the same age and species, where dead wood is removed, where understory is suppressed, support far less biodiversity because they eliminate most of the environmental variation that creates niches for different species. I leave the stream behind and climb a gentle slope leading back toward the main trail, passing through a section of forest dominated by big leaf maples. These deciduous trees create different conditions than the conifers. Their leaves decompose more quickly, creating a different quality of leaf litter, and they shed their leaves in autumn, creating pulses of resource availability that structure decomposer communities differently than the steady input from evergreen conifers. The fungal communities here reflect these differences. Some species are generalists occurring under both hardwoods and conifers, but many are specialists associated with particular tree types. The ground beneath the maples is carpeted with fallen leaves in various stages of decomposition. Fresh tanned leaves that fell recently, darker leaves from previous years partially broken down, and beneath those, a layer of fragmentary leaf material transitioning into soil. If I were to examine this leaf litter microscopically, I'd see it teeming with fungal hyphae, bacteria, protozoa, nematodes, and countless tiny arthropods, all participating in the decomposition process. The transformation of a fallen leaf into soil humus involves thousands of organisms working in complex succession, each contributing to breaking down different compounds. Leaf decomposition follows predictable stages. Initially, soluble compounds like sugars and amino acids are rapidly leached out by rain and consumed by bacteria and fungi. This is the fast phase, occurring over days to weeks. Next comes decomposition of cellulose, the structural polymer that makes up much of leaf material. Many fungi can break down cellulose using cellulase enzymes, but the process is slower than consuming simple sugars, taking weeks to months. Finally comes lignin decomposition, the slowest phase, requiring specialized fungi, primarily white rot and brown rot fungi, that produce powerful oxidative enzymes capable of breaking lignin's complex chemical bonds. This phase can take months to years. The rate of decomposition varies dramatically with climate, substrate quality, and decomposer community composition. In tropical rainforests, a fallen leaf might decompose completely within weeks, while in cold or dry environments, the process might take years. Here, in the temperate Pacific Northwest, with its mild temperatures and abundant moisture, decomposition is relatively fast. Most leaves are substantially decomposed within a year, though lignin-rich conifer needles take longer than hardwood leaves. I notice a small patch of snow-white mushrooms pushing up through the maple leaf litter, probably Mycena species, delicate and ephemeral, likely to survive only a few days before collapsing back into the leaf litter themselves, completing their brief mission of spore dispersal before their own tissues are decomposed by other fungi and bacteria. This cycle, organisms decomposing dead material, then themselves becoming dead material to be decomposed, is the fundamental engine of nutrient cycling that allows ecosystems to persist. Without decomposers, nutrients would remain locked up in dead organic matter indefinitely, unavailable to plants, and ecosystems would grind to a halt as available nutrients were depleted. Fungi, along with bacteria, are the key that unlocks this material, breaking chemical bonds that require energy to form and releasing nutrients back into circulation. In a very real sense, decomposition is the flip side of photosynthesis. Photosynthesis uses solar energy to build complex organic molecules from simple inorganic ones, while decomposition breaks those complex molecules back down into simple ones, releasing the energy as heat and making nutrients available for new growth. The slope levels out, and I find myself in a small natural clearing where a massive old stump dominates. All that remains of a tree that must have been enormous before being cut, probably a century or more ago, based on the stump's advanced decay. The stump is a universe unto itself, supporting dozens of plant and fungal species. Mosses cover much of its surface, small ferns grow from cracks, and mushrooms erupt from every side. The wood is soft and punky, easily penetrated by a finger, shot through with mycelial threads and insect tunnels, more air than solid wood at this point. This stump will continue to provide ecological services for decades more before it completely merges with the soil. It acts as a seedbed for trees. I can see several small hemlocks and a Douglas fir seedling growing from its surface, their roots penetrating into the decaying wood. It's a reservoir of moisture and nutrients, slowly releasing both as it decomposes. It provides habitat for invertebrates, salamanders, small mammals, and countless microorganisms. And it serves as a monument to the forest's history, a reminder that this landscape has been logged and is now regrowing, healing from past disturbances. Looking at this old stump makes me think about forest time scales and human time scales and how poorly they align. From a human perspective, a century seems long. It spans multiple human lifetimes. But from a forest perspective, a century is relatively brief, just one phase in the ongoing cycle of growth, death, and regeneration. The tree that produced this stump probably lived 3 or 4 centuries before being cut. The stump has been decomposing for a century and will continue for decades more. A new tree growing on this site might live 3 centuries, then decompose for a century, then support new growth again. The cycles are long, patient, beyond the scale of individual human experience, yet humans are now affecting forests at scales and speeds that have no precedent in forest history. Climate change is altering growing seasons, shifting species ranges, changing fire regimes. Introduced pests and diseases are causing widespread mortality. Dutch elm disease, chestnut blight, sudden oak death, white pine blister rust, and countless others. Habitat fragmentation is isolating populations and disrupting ecological processes. Logging continues at scales that prevent many forests from developing old growth characteristics. The question facing forest ecologists is whether these forests can adapt quickly enough to the rapid changes we're imposing, or whether we're pushing systems beyond their capacity for resilience. Fungi might be part of the answer. Their rapid reproduction, enormous genetic diversity, and capacity for adaptation mean they can potentially respond more quickly to changing conditions than the trees they partner with. Mycorrhizal networks might help forests respond to stress by redistributing resources from less affected to more affected areas. Fungal decomposers might speed nutrient cycling in ways that help forests recover from disturbance. But fungi face their own challenges. Some species have narrow habitat requirements and may not be able to disperse to new suitable areas as climate shifts. Others might be disrupted by the fragmentation that affects their host plants. I'm approaching the final stretch of trail that will lead me back to the parking area, but the forest continues to offer discoveries. A large cluster of honey mushrooms catches my attention. These are probably from the same mycelial individual I saw earlier in the day, connected underground by rhizomorphs, root-like structures that transport nutrients and allow the fungus to colonize new territories. Rhizomorphs can extend for meters through soil, seeking new host trees or substrates, and they help explain how some fungal individuals can grow to such enormous sizes. The honey mushroom I'm observing is both fascinating and controversial. As I mentioned earlier, Armillaria species can be severe forest pathogens, killing valuable timber trees and ornamental species. But they're also important decomposers, and play natural roles in forest dynamics. The key is context. In managed forests, where every tree has commercial value, honey mushrooms are pests. In natural forests, where tree mortality is a normal part of forest cycles, they're simply one component of a complex system. This raises important questions about how we perceive organisms as good or bad, beneficial or harmful. These judgments usually reflect human interests rather than ecological roles. From a forest ecosystem perspective, there's no such thing as a pest. Every organism has evolved to exploit available niches and resources. Tree-killing fungi create canopy gaps that allow regeneration, provide dead wood that supports decomposer communities, and cycle nutrients back into the system. They're only bad from the narrow perspective of maximizing timber production or maintaining ornamental landscapes. The same is true for many organisms we label as pests or invasives. Some genuinely cause severe ecological disruption. Chestnut blight essentially eliminated American chestnut as a canopy dominant in eastern forests, fundamentally altering ecosystem structure and function. Others, however, are incorporated into ecosystems without causing collapse. They become novel species in a reconfigured community. The challenge is predicting which outcome will occur and managing the former while accepting the latter. I notice the light has taken on the warm, golden quality of approaching sunset, filtering through the canopy in shafts that illuminate floating spore clouds. Millions of microscopic propagules drifting on air currents, most destined to land in unsuitable locations, but some, by chance, reaching perfect conditions for germination and growth. This is the visible reminder of the invisible spore cloud that surrounds us always. We're breathing fungal spores with every breath, their concentration varying with season, weather, and location, but always present. For most people, this constant inhalation of fungal spores is harmless or even beneficial. Exposure to diverse microbes helps train our immune systems. But for some, fungal spores trigger allergies or asthma. And for immunocompromised individuals, some environmental fungi can cause serious infections. The opportunistic pathogens that normally live benignly in soil or on plants can become dangerous when they encounter hosts with weakened immune defenses. This is why hospitals take air quality so seriously, filtering air to remove spores and other particles. But the vast majority of fungi have no interest in infecting humans. We're too hot. Our body temperature of 37 degrees Celsius is above the thermal tolerance of most fungi, which evolved to thrive at the cooler temperatures of soil and wood. This temperature barrier is actually one of our major defenses against fungal infection. The fungi that do infect humans are either adapted to mammalian body temperature or infect superficial tissues like skin and nails where temperature is cooler. As climate change raises global temperatures, there's concern that some environmental fungi might adapt to tolerate higher temperatures, potentially becoming new human pathogens. I'm almost back to the trailhead now, emerging from the deep forest into an edge zone where more light penetrates. Edge habitats have their own characteristic species. Some fungi fruit more readily in open areas with higher light and temperature, while others prefer the stable conditions of forest interiors. The mushroom diversity in edges can be high because these zones combine elements of both open and closed habitats, creating more niches than either habitat alone. The trail passes by a small educational sign about fungi that the Land Management Agency has installed, with basic information about mushroom identification safety and ecological roles. I appreciate these efforts to educate the public about fungi, though I wish the information went deeper into the sophistication of fungal ecology. Most people's knowledge of fungi is limited to mushrooms in grocery stores, molds on forgotten food, and vague awareness that some wild mushrooms are poisonous. The incredible ecological importance of fungi, their evolutionary relationships, their biochemical sophistication, all of this remains unknown to most people. Part of the challenge is that fungi are largely invisible. Unlike plants and animals that we can easily observe, most fungal activity happens underground or inside substrates, out of view. Mushrooms provide temporary visibility, but they're just the reproductive structures, not the organism itself. It's like trying to understand an oak tree by only ever seeing its acorns. You'd miss the massive trunk, the spreading branches, the photosynthesizing leaves, the interconnection with other trees through root grafts and mycorrhizal networks. But awareness is growing. Books about the wood wide web and fungal networks have reached mainstream audiences. Documentaries about fungi are attracting viewers beyond specialist communities. Citizen science projects like iNaturalist allow people to photograph and share mushroom finds, contributing to scientific understanding of fungal distribution and phenology while learning about identification themselves. There's a growing fungal appreciation movement, people recognizing that these organisms deserve attention and respect for their crucial ecological roles. As I reach my vehicle in the parking area, I take one last look back at the forest, now shadowy in the failing light. Somewhere beneath those trees, the mycelial networks I've been contemplating continue their work, unaffected by day or night, moving nutrients, supporting trees, decomposing wood, doing the work that makes forests possible. The mushrooms I observed today might collapse and disappear in the next rain, but the mycelia that produce them will persist, continuing to grow, continuing to fruit when conditions are right, continuing the ancient partnership between fungi and forests that has shaped terrestrial ecosystems for hundreds of millions of years. Driving away from the trailhead, my mind continues to process the day's observations, fitting them into the larger framework of ecological understanding. The forest I just walked through isn't a static collection of organisms, but a dynamic system in constant flux. Trees growing and dying, fungi fruiting and decomposing, nutrients cycling from soil to plant to decomposer and back to soil, energy flowing from sun to photosynthesis to consumer to decomposer to heat. Everything interconnected, everything in motion, everything part of patterns that operate on timescales from seconds to millennia. I think about the mycorrhizal networks that connect those trees and how recent this understanding is. For most of human history, we saw trees as individuals competing for resources. Then we recognized that they also cooperate, sharing resources through root grafts. Then we discovered mycorrhizal partnerships and initially understood them as simple bilateral relationships. One tree, one fungus, trading nutrients for sugars. Only recently have we recognized that the networks are vastly more complex. Single fungal individuals connecting to multiple trees, single trees connected to multiple fungi, resources and signals flowing through the network in patterns we're only beginning to map. This evolving understanding illustrates how science works, not as a collection of fixed facts, but as an ongoing process of discovery and revision. What we know about fungi today will be refined and expanded by future research. Some current ideas will be confirmed, others modified, some perhaps overturned entirely. This doesn't mean current understanding is worthless. It's the best explanation available given current evidence. But it does mean we should hold our knowledge with appropriate humility, recognizing its provisional nature. The fungal world is full of unsolved mysteries. We still don't fully understand how mycorrhizal networks regulate resource flow, whether it's passive diffusion driven by concentration gradients or active allocation controlled by the fungus or trees. We don't know how trees recognize kin through mycorrhizal networks, what signals they exchange, whether fungi manipulate these exchanges to their own benefit. We're unsure about the degree to which different tree species compete versus cooperate through shared networks. These questions are the frontier of forest ecology, being investigated through clever experiments, but definitive answers remain elusive. Part of the challenge is that these systems are incredibly complex and difficult to study. You can't easily track nutrient movement through underground networks spanning thousands of square meters. You can't observe the system without disturbing it. Digging to access roots and fungi fundamentally changes the conditions you're trying to measure, and the timescales involved, years to decades for some processes, make experiments difficult to conduct within the career span of individual researchers or the funding cycles of grant agencies. Yet progress is being made through careful, patient work. Isotope tracer studies inject distinctive forms of carbon or nitrogen and track where they appear, revealing flow patterns through networks. DNA analysis identifies which species are present and how they're distributed. Experimental manipulations, severing mycorrhizal networks, removing competing trees, adding resources, reveal how systems respond to changes. Slowly, incrementally, the picture comes into focus, though many details remain fuzzy. I arrive home as darkness settles completely, my mind still dwelling in the forest, still processing observations and questions. Inside, I spread my notes and photographs on the table, beginning the process of identifying the specimens I documented. Some are straightforward. The honey mushrooms, chanterelles, and fly agaric are distinctive enough for confident identification from field characteristics. Others require careful comparison with field guide descriptions, checking multiple features to narrow down possibilities. Several specimens resist identification. They have characteristics that don't quite match any species in my guides, or they match several species that require microscopic examination of spores to distinguish. This is the reality of mushroom identification. Unless you're willing to collect specimens and examine them microscopically, many will remain identified only to genus or group. And even with microscopy, some require DNA sequencing for certain identification. Fungal taxonomy is challenging, with many look-alike species, regional variation, and ongoing taxonomic revision as DNA analysis reshapes our understanding of fungal relationships. I make notes about habitat associations, careful to record not just what mushrooms I found, but where, what trees were nearby, what substrate they were growing on, what other plants were present, what the microhabitat conditions were like. These ecological details are as important as morphological features for understanding fungi. A mushroom's identity is intimately linked to its context. Many species are so specifically associated with particular hosts or substrates that knowing where you found it narrows identification considerably. Looking through my photographs, I'm struck again by the aesthetic beauty of fungi, the elegant curves of mushroom caps, the delicate structure of gills, the vibrant colors ranging from pure white through yellows, oranges, reds, purples, browns, blacks, and every shade between. Evolution has sculpted these forms to maximize spore dispersal efficiency, but in doing so has created structures that humans find beautiful. Perhaps this isn't coincidental. Perhaps our sense of beauty is partly tuned to recognize patterns that indicate health, fertility, complexity, the qualities that make ecosystems and organisms successful. Or perhaps I'm overinterpreting, projecting meaning onto forms that just are, products of physical and biological processes operating without aesthetic intention. A mushroom isn't trying to be beautiful. It's trying to disperse spores. That we find it beautiful says more about us than about the mushroom. Yet the beauty is real, a quality that emerges from the interaction between object and observer, neither purely subjective nor purely objective, but created in the space between. I think about the invisible networks beneath the forest floor, extending in 3 dimensions through soil, connecting organisms in ways we can't directly perceive. We live in a visible world. Our senses evolved to detect what's above ground, in the light, directly observable. But the underground world is vast and complex, teeming with life and activity, crucial to everything we can see but largely hidden from view. Fungi help bridge that gap, producing visible mushrooms that hint at the invisible mycelial networks below. As the evening deepens, I prepare a simple meal, including some cultivated mushrooms from the market, not the wild specimens I observed today, which I left in the forest, but domesticated varieties bred for flavor and reliable cultivation. Shiitake, originally cultivated in China and Japan for over 1,000 years, now grown worldwide on logs or sawdust blocks. These cultivated mushrooms are genetically removed from their wild ancestors, selected for traits like rapid growth, high yield, resistance to contamination, and consistent fruiting. Mushroom cultivation is an ancient art that has become a modern science. The basic principle is simple. Provide the fungus with appropriate substrate, temperature, humidity, and light conditions, and it will produce mushrooms. But the details are complex, varying enormously between species. Some mushrooms, like oyster mushrooms and shiitake, are relatively easy to cultivate and have been domesticated thoroughly. Others, like morels and matsutake, resist cultivation despite decades of attempts, their requirements either too specific or still poorly understood. The global mushroom industry is substantial, producing millions of tons annually, with white button mushrooms and their close relatives, Cremini and Portobello, which are actually the same species at different maturity stages, dominating production. But specialty mushrooms are growing in popularity as people develop appreciation for diverse flavors and textures. Oyster mushrooms with their delicate flavor, lion's mane with its seafood-like texture, maitake with its complex taste. Each offers something unique, and cultivation allows year-round availability regardless of natural fruiting seasons. Yet, something is lost in cultivation: the connection to place, to ecosystem, to the complex relationships that wild fungi maintain with their environments. Cultivated mushrooms grow on sterilized substrate in controlled conditions, divorced from the forest floor, from associated plants, from the community of organisms that surrounds them in nature. They're optimized for human consumption, but they're no longer fully themselves, no longer participants in nutrient cycling, no longer connected through mycorrhizal networks, no longer fulfilling ecological roles. This tension between cultivation and wildness applies to many foods. Cultivated vegetables are nutritious and reliable, but they're genetic shadows of their wild ancestors, selected for traits we value: size, yield, shelf life, often at the expense of nutritional density, flavor complexity, or ecological adaptation. Wild foods, foraged mushrooms, berries, greens, connect us more directly to the land, to seasonal rhythms, to the skill of identification and the knowledge of place. But they're less reliable, require more effort to obtain, and carry risks that cultivated foods don't. Perhaps the answer isn't choosing between cultivated and wild, but appreciating both for what they offer. Cultivated mushrooms provide reliable nutrition and pleasure without ecological impact. Wild mushrooms carefully foraged following sustainable practices, offer deeper connection to ecosystem and place, along with unique flavors that cultivation can't replicate. Both have their place in a balanced relationship with the fungal world. As I eat, I review my field notes from the day, adding details I remember, sketching diagrams of particularly interesting specimens, noting questions for further research. How do the cordyceps I read about find their insect hosts? What determines which trees become connected by mycorrhizal networks and which remain more isolated? Why do some years produce abundant mushroom flushes while others see few fruiting bodies despite seemingly similar conditions? Each observation generates questions. Each answer suggests new investigations. This is the nature of naturalist study, not just collecting facts, but developing deeper understanding through repeated observation, pattern recognition, question formulation, and attempted answers. The goal isn't comprehensive knowledge, that's impossible given the complexity of natural systems, but rather increasing appreciation for that complexity, growing awareness of relationships and processes, developing intuition about how ecosystems function. I think about returning to that forest in different seasons. Winter, when few mushrooms fruit but fungal activity continues underground. Spring, when morels might appear in disturbed areas and the first mushrooms emerge as soil warms. Summer, when different species fruit in the warmth. Each season offers different insights, different species, different aspects of the annual cycle that fungi navigate. Understanding requires observation across seasons and years, building a picture that includes temporal variation, not just spatial patterns. As night settles completely outside my window, I feel a deep satisfaction with the day, the physical exercise of hiking, the mental engagement of observation and identification, the spiritual connection to forest ecosystems that comes from paying attention, from trying to understand rather than just passing through. The forest will continue its work whether I'm present or not, whether I understand it or not. But my understanding enriches my experience and, I hope, makes me a better steward of such places. Fungal networks will continue their patient work tonight, growing, transporting nutrients, forming new connections, supporting trees, decomposing wood, cycling nutrients that allow the forest to persist. Mushrooms that fruited today will begin to collapse, their spores already dispersed, their tissues soon to be food for other organisms. New primordia, mushroom initials, are forming underground, waiting for the right conditions to emerge. The cycle continues, as it has for hundreds of millions of years, as it will for millions more if we protect the ecosystems that support it. I think about the responsibility that understanding brings, knowing how crucial fungi are to forest health, how sensitive they are to disturbance, how long it takes for complex fungal communities to develop. This knowledge creates obligation to protect and preserve. Not all land can or should be locked away as wilderness, but enough must be protected to maintain functioning ecosystems, to preserve the full complement of species and relationships, to allow natural processes to continue. Climate change, habitat fragmentation, invasive species, pollution, all pose challenges to forest ecosystems and the fungal communities they depend on. But fungi's adaptability, their genetic diversity, their rapid reproduction, might make them resilient if we give them the conditions they need. Protecting old-growth forests, maintaining deadwood, limiting soil disturbance, preserving habitat connectivity, these practices support fungal diversity and, through fungi, support entire forest ecosystems. As I prepare for sleep, my mind returns to the mycelial networks extending through soil, the hidden architecture that supports visible life. In the darkness, the forest continues its ancient rhythms, Photosynthesis paused until dawn, but decomposition continuing, nutrients flowing, fungi growing, life persisting in forms both visible and invisible, both active and dormant, all interconnected in the vast web of relationships that makes ecosystems possible. The mushrooms I observed today were just glimpses, brief windows into a hidden world, temporary expressions of organisms whose main existence happens out of sight. But those glimpses are enough to inspire wonder, to provoke questions, to drive the curiosity that leads to understanding. Tomorrow, the forest will be slightly different. Some mushrooms collapsed, others newly emerged, the mycelial networks reconfigured in subtle ways, the endless cycle continuing. And I'll return, when seasons shift and conditions change, to observe again, to learn more, to deepen my appreciation for the fungal kingdom and its essential roles in the living world. Each visit adds to understanding. Each mushroom observed contributes another piece to the puzzle. Each season reveals different patterns. The study of fungi is endless because the subject is vast, complex, and constantly changing. Not a body of facts to memorize, but a living system to engage with, to wonder about, to protect. The forest teaches patience. Processes that take decades or centuries can't be rushed, can't be fully understood without sustained observation. It teaches humility. The more you learn, the more you realize how much remains unknown. It teaches interconnection. Nothing exists in isolation. Everything affects and is affected by everything else. And it teaches gratitude for the opportunity to observe, to learn, to participate, however briefly, in systems far older and larger than ourselves. Sleep comes gently, my mind still wandering through the forest, still seeing golden chanterelles pushing through moss, honey mushrooms clustered on logs, networks of mycelium extending through darkness and soil, connecting all into one vast living community. The invisible made briefly visible, the hidden revealed in momentary fruiting, the patient work of decomposition and partnership continuing forever beneath our feet, supporting the green world we see, making possible the forests and all the life they contain. Thank you for joining this journey through autumn forests and fungal networks, exploring the hidden connections beneath our feet and the quiet organisms that make forests possible. If these stories help you find peace, perspective, and restful sleep, please consider subscribing to our channel. Each tale we share is an invitation to slow down, to appreciate the intricate beauty of natural systems, and to drift into dreams feeling connected to the living world. May you sleep well, dream of forests and mycelial highways, and wake with renewed wonder for the complexity that surrounds us. Good night, and thank you for exploring the fungal kingdom with us.